Good afternoon, students. Instead of live class, I can send this video. So this record class is very useful. I can continue the last live class. We have discussed about that what is DPT, what is pi, everything, turgid, everything. Today we will go and discuss osmosis. What is osmosis? This is a Latin word impulse, urge. So it is a special type of diffusion. Is one of the method is diffusion. So it represents the what it will be represent water or solvent molecule through a selective permeable membrane from the place of the higher concentration to lower concentration. So that is a higher water potential to lower water potential. This is the basic one. If you know very well means in our school, college, in our home we can use this osmosis method only for filtering the water. Okay, so what is the types of the solution we can use in the concentrations? Here we are using the hypertonic or hypotonic. Hypo means low, I already told, and hyper means high. This is a strong solution which attract the solvent. Okay, and the hypotonic, it diffuse water out to other solution. So this is a very important. Next, isotonic. So we can water filter we can use in our home. This method only the water filter and we can use it and get that pure water. Okay, pure water we can get in this method. Why? So we can add a dirty water or whatever it is that is having the higher concentration. After that it will be everything we can take it from the semi permeable membrane, it attract, and after that we are getting the solution is a pure method. So higher concentration to the lower concentration. Maybe it is a lower concentration to higher concentration. So for the salt water we can use it, and after that we are getting in the new water, pure water means this is the base one. Okay, isotonic. Iso means identical. Tonic means solute. Okay, not a salt. It is a wrong solute. That is the meaning. It refers to the two solution having a same concentration. In this condition, movement of the water molecule will be zero. The term hyper or hypo isotonic, it is a which can be used only in the another solution only. Okay, so this is the important one. Tisiphonal experiment we can do in our laboratory. So only for the experiment only, demonstration only, not we don't want to think about that, we don't want to do that. Okay, so see this is the picture represent that it's a hypotonic low, hypertonic it is a high concentration. So hypo means inside coming and hyper means it will be outside going. Okay, so this is the method. So already we have discussed it is what is the pi and what is that zero and solute. So see that the pi value is pi of the water is 0 and the potential also 0, solute also 0. So the same way that water having the solute and everything it is the same only, but especially in the potential it is a 0 only. Okay, another thing is the same 5. Okay, so next we go for the plasmosis. What is the plasmolysis? So breakdown. Lysis means breakdown. The plasma means cytoplasm is kept in the hypotonic solution and the waters are leave in the exoosmosis. Okay, outside. Okay, exo means outside, endo means inside. So shrink and cell membrane, it's pulled away from the cell wall and finally the cell become a flaccid. This process name is the plasmolysis. No doubt this is a 2-mark question, important question. What is plasmolysis or define plasmolysis means we can explain this. Okay. Next, wilting of plants noticed under the condition of water scarcity is one indication of plasmolysis. So how many types of the plasmolysis is that? There are 3 plasmolysis: incipient, evident and a final plasmolysis. Okay, these are the 3 types of plasmolysis. What is the importance of this plasmolysis? So only it is living cells, and so it is used to test whether the cell living or dead. Okay, so next we go for the deplasmolysis. The effect of plasmolysis can be reversed by the transferring the back into the water hypotonic solution. So due to the endomesis, so again the target it will be regained. So this is the getting from the original shape. This is only diplolysis. So example we can impression of the dry resin in water. So potato osmoscope, it is very important one. So this is one of the practical I can explain now. You can understood, and after that if you have a doubt means extra. Next live class you can ask. Okay, what we will do this demonstration only. It is also demonstration only. Don't want to think about that. You don't want to do in the practical lab exam. Okay, demonstration what will you can take the first potato. Okay, then you can peel the potato and we can cut into this side. Okay, with the use of the knife. Fill the cavity with concentration sugar solution and then we can mark the initial level. Then we can place the setup beaker of the pure water. Okay, after 10 minutes observe the sugar solution level and record your finding. So this is the why we can help of your teacher in discuss result means we can do it in our laboratory. So this instrument we can, so what is that, we can take a beaker and peel it and we can cut in this shape, and first we can add the sugar solution into that and we can mark that, and after that we can think about the after 10 minutes for a few minutes it will be increase the level of the water. Why it is happening? Because it is the happen from the diplomacies. This is the one of the example of deplasmolysis. What is that? The meaning is that, so after that getting the hypotonic solution, in the because of the endomyosis the cell become turgid again. Okay, next we go for the reverse osmosis. Reverse osmosis follow the same principle of the osmosis but in the reverse direction. Okay, the same only. It is only applying the water against the concentration gradient of the solution. So it is a regular osmosis. The water molecules move from the higher concentration to the lower concentration. Okay, so pure water to salt water. But it is a reverse osmosis. What will happen in that? Lower concentration to higher concentration. That means salt water to pure water. Okay, this is the, we can use the reverse osmosis. Why it is used for the, where it is used, that I already told, purification of the drinking water. In this method only we are using. Okay, desalination of seawater. Okay, desalination, we can take it all the salt content from that water. This we call as a desalination of the seawater. This is a method of reverse osmosis. Okay, next, what is the difference between the plasmolysis type? Incipitants, evident and final plasmolysis. No morphological symptoms. Here a wilting of the leaf appear, severe wilting and drooping, the plasma membrane separate and the plasma membrane completely detached from the evident plasmolysis. Final plasmolysis, it is a wall maximum shrinkage of the volume. It is reversible. 3 of them, it is a reversible one thing only. So next, absorption of the water from soil to root hair either actively or passively. Okay, from root hairs further transport to the later side, and then direction of the xylem, it is a super highway of the water transport. Okay, next, water absorbing organ. Usually absorption of the water occur in plants through the young root. The zone of the rapid water absorb in the root hairs. Okay, so it is the surface of the absorption only it will be happen. So there, what are the way in the water across the root cells means, there is a way is there where the root hair first it absorbed and after the epidermal cells, so outside of the cell, otherwise we call as the thermal skin of the plant. Then centripetally across the cortex and pericycle and finally reach into the xylem. Okay, the method of osmotically. There are 3 types that is possible, or 3 routes is possible. There is a way, no? So what are they? Apoplast, sympaplast, transmembrane route. Okay, what is apoplast? This is the away. Plast means cell. Okay, apo means away. Sympa means within, inside the cells, and the transmembrane means we can see it in the transmembrane and where it is crosses, so that way we can see that. Okay, next we go for the mechanism of water absorption. So there are absorption, how it is active absorption and passive absorption. There are 2 types of absorption is there. First they told that grammar in the year of the 1949 there are 2 distinct mechanism is there. So active absorption means, what is that means, water absorbed due to the force generator in the root itself, that is we call as a active absorption. Okay, then osmotic active absorption, there are theories that say for Alkins and Priestley. Okay, Alkins say in the 1916, Priestley is the 1923. According to this theory, the first set of absorption is soil and then it is embedded by the cell of the root hair, then followed by the osmosis. What and all already we have discussed the same only, but it is in the soil and the root pressure also important one. So what are the objection of this osmotic theory means? So there are xylem is not always high, root pressure is not a universal. These 2 only objection. Next, non-osmotic active absorption is Bennett, Clark and Diamond and Kramer. Kramer also they already say there are absorption. Okay, grammar only, mechanism water absorption told. So it is based on that observation of osmotic theory, the cell sap concentration xylem is not always high, root pressure is not universal in all plants, especially in the trees. Okay, next non-osmotic active absorption and passive absorption. Non-active means nothing but it is a ATP and synthesis with the lower concentration of the soil water, then it is a high movement expenditure is needed. Next, passive absorption. The passive absorption route do not play any role in the absorption of the water, but it is a transpiration only. It is regulated the transpiration only, and then drop of the turgor pressure, then increase the DPT. So this is the passive absorption. So already table 11.3 the passive absorption is shown, we can discuss. Next, concept map, movement of the water in the osmotic system based on the various parameter, low zero solid concentration, high solvent concentration, hypotonic, high osmotic potential. And the low osmotic pressure is the pure water, salt water. So based on this map only the active result happen in that. Okay, so active absorption and passive absorption, 3 mark question, mark down. So what is the difference between active, passive and the absorption? Here active absorption is a take place by the actively route, and the pressure of the absorption is not developed by the passive route. Okay, here active absorption regulated that, no effect of that, here regulate that. In the DPT is root hairs have a always high, here is a tension created xylem sap, here reabsorption energy needed, here respiration energy is not required, here symplastic movement of water, here apoplastic movement of water is involved. Okay, so next class we can see about the what is the balsam plant and experiment. This is also only theoretical only, so don't worry about that. So next class we will discuss in this chapter. Okay, so thank you students.